name is Evergren. My research is focused on understanding how cells communicate, both to the outside environment and internally. We're a cell biology lab that investigates how cells can import cargo and traffic both membranes and molecules. These are necessary mechanisms for a cell to communicate and to react to its environment in an appropriate way. We're located in the Patrick G. Johnston Centre for Cancer Research at Queen's University Belfast. In our lab, we use both cell and molecular approaches to investigate mechanisms of membrane trafficking. Obstruction of membrane trafficking in the cells can affect downstream functions and contribute to driving cancer progression. Our research focuses on investigating the molecular mechanisms that underpins subcellular trafficking within cells. These pathways are critical for regulating cell signaling by receptors at the cell surface, internalization of nutrients, energy homeostasis, cell viability, and proliferation. Recently, we showed that these mechanisms are also regulating cellular cannibalism, which is a critical mechanism for cells to cluster together and form subgroups with specialized functions. In this context, the cells that by default randomly stick together can move away from each other by taking a bite out of its neighbour and digesting it, thereby detaching itself and being able to move away and group together with similar cells. Here we analyse the patterns that cells form before and after we had perturbed molecular interactions that are important for membrane trafficking. This research identified novel interactions and players that facilitate cellular cannibalism. In addition, we are interested in investigating the role of membrane binding proteins in novel functions such as membrane constriction, membrane contact site formation, and stress responses. In this example, we were able to describe a new mechanism for how cells can pinch off membranes. In our research, we use a range of different techniques, from biochemistry to light cell imaging. Together, these approaches provide us with a unique set of tools to give new insights into membrane trafficking and how these events become dysfunctional during pathogenesis, such as cancer. One research project that we are working on is investigating the role of post-translational modifications in receptor-mediated internalization and trafficking. In this experiment, we took short videos of cancer cells. Each dot that flashes represents a receptor carrying nutrients that is internalized by the cell. Once it has been imported in a membrane bud, the light disappears. In the control cell, you can see that it happens very rapidly. To another set of cells, we applied a drug that inhibits an enzyme that facilitates post-translational modifications of proteins that impact on membrane trafficking. And you can see that this had a dramatic impact. It slows down the trafficking right down, and you can see how static the points are. This is important, for example, because cancer cells have a high metabolism and are dependent on the import of nutrients. These mechanisms are thus critical to sustain both cell survival and proliferation. We also use human prostate cancer tissue in collaboration with Professor Lisa Butler at the University of Adelaide to investigate the impact of drugs used in the clinic on membrane trafficking to see how organelle contact with another within the tumor tissue changes in response to a drug. This research aims to discover new therapeutic targets which can be exploited to develop new therapy strategies for prostate cancer patients in the future. It's a challenge for us to continue our lab research during the COVID-19 pandemic. During this time, I have been and continue to work from home, where I write scientific papers, for example. It has given me the opportunity to take a step back and reflect on the results that we have been generating to come up with new ideas and hypotheses to test in the future. I've also been writing grant applications to apply for funding to continue our research that we hope long-term will benefit cancer patients.